Okay, so welcome everyone. It's my pleasure today to welcome to BU, but also to introduce all of you to one of my close colleagues, Dr. Barb Triggs-Raine. People who have taken my courses have probably heard of her multiple times, so at least the name should not be the name should be quite familiar to you. So Dr. Triggs-Raine is a full professor in the Department of Biochemistry and Medical Genetics. So that is at the Health Sciences Center at the University of Manitoba. And it's not in the little bio that I have here, but she just stepped down is maybe the wrong term, retired, phased out as department chair after really rejuvenating a department and hiring a whole bunch of new people into that department, which makes it quite an active and vibrant place, which is really to her credit for that. She is truly a Manitoban through and through. She did her undergrad and her master's and her PhD all at the University of Manitoba, namely in microbiology. And then she went on to postdocs at both Sick Kids and at the Montreal Children's Hospital in medical genetics or what sometimes could be called clinical genetics. She is a PhD, not an MD, but kind of bridges that field of rare genetic diseases and especially trying to identify the genetic variants that cause these rare genetic diseases. The bio here is a bit interesting, and I have to agree with this, and I'm reading it directly. The late 80s was an inspiring time for Canadian geneticists who were leading the way in the discovery of many disease genes. As she remembers well the celebrations when the cystic fibrosis gene was first identified at Sick Kids. And I think that that's quite true, right? Canada has a long-standing history or tradition, you may not realize this, of like extraordinary excellence in genetics and specifically in trying to identify these the causes of these rare genetic disorders. Fortunately for us, the University of Manitoba was able to recruit her back in 1991, where she became a assistant professor and rose through the ranks in the Department of Biochemistry at the medical school again. And I was teasing her a little bit here a second ago in probably saying that she's identified the genetic basis of more rare genetic diseases than anyone else that I can think of. So she's truly a giant in the field. So it's really a pleasure to have her here. Basically, in chatting with Dr. Majunder this year, we kind of thought, oh, we really should invite her. She is going to be talking about something that you've all heard of. I'm not going to give her thunder away, but some of the genetic causes that she's identified for this genetic disease that I know you've all seen, but maybe not thought of before. So Dr. Triggs-Raine, I'll let you take it away.
Okay, well, thanks so much, Mike, for the really generous introduction. And it's such a pleasure to be here with you today. I'm sorry that I'm not there in person. When I first got up this morning, there was a lot of snow, and as the day went on, it looks a little better, and I thought, oh, maybe I could have gone, but hopefully I'll get there again in the future. So thanks so much for having me today. I am going to talk about, I can't remember exactly which things I've shared with your class before, Mike, but today I entitled my talk, Sticky Business, the Crucial Role of Hyaluronan Breakdown for Normal Development. And so I'll be talking about a disorder of hyaluronan breakdown in my talk today. But before I continue with that, I'd like to do a traditional territories acknowledgement. I'm sure that you typically do this in Brandon as well. I've been really lucky, as Mike pointed out, to work for 35 years at the University of Manitoba. And those campuses are located on the original lands of the Anishinaabe, the Niniwak, the Niseneniwuk, the Dakota Ayote and Dene, and the national homeland of the Red River Métis. And we respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. And then kind of keeping with that approach, I'd just like to say a little bit about where I actually originated, and that was from a farm about halfway between Brandon and Winnipeg. So when I grew up between Holland and Triggs-Raine, in case any of you have heard of that, and please, by the way, it's hard for me to keep track of what's happening in the classroom. So if you at any point want to interrupt today and say anything, just yell it out, and I'm comfortable with that. But I want to show you where I came from. We often traveled to Brandon to shop and for events from school. And for some strange reason, I never got the chance to see Brandon University during those times. So I'm still holding out for that. I'll definitely make a point of coming by to visit at some point in the future. So maybe I'm going to start, as I said, I can't see everybody in the classroom, I don't think. But just shout out if, have you heard of hyaluronan?
No.
No? Okay. Well, hopefully after this, some of you have?
Well, some of the people who've taken my class in the past where we did your topic, they have.
Well, if you haven't heard of hyaluronan, I have this feeling that when you go to the Shoppers Drug Mart or whatever grocery stores you go to, that you're going to notice that hyaluronan is in like everything. You know, if you pick up a moisturizer or a makeup off the shelf these days, I don't think it's got any kind of class unless it's got hyaluronan in it. And they don't just put this stuff in your moisturizer. It's on the coating of tubes that are used in the surgeries, on bandages to prevent adhesions, and almost every eye drop that's presented for dry eye disease has hyaluronan in it. And if you know anything about its structure, you'll know that it binds a lot of water, so it's really good for moisturizing things. And so that's part of why hyaluronan is used in all these creams. But also, they have used hyaluronan as an injectable for a long time, and no one ever makes any antibodies against it. So it's really great for medical applications. And some of the first medical applications of hyaluronan were actually when it was injected into joints of racehorses. If you have osteoarthritis, as some racehorses apparently do, hyaluronan was thought to be able to provide some relief to the pain that was associated with your bones rubbing against each other because they didn't have any cartilage left, which is what happens in osteoarthritis. And so whether this really works well is really a bit up in the air now, but it is a billion-dollar business. It's not just in the joint, though, that they inject hyaluronan. It's often injected to provide a scaffold in surgeries for cells to grow on. So let's say you have a little leak in the bladder. You might have a bit of hyaluronan put up so that cells can grow around it. And maybe the most popular use is as a cosmetic filler. So people get all kinds of injections of hyaluronan as part of cosmetic applications to fill wrinkles and to plump lips and things like that. It's not all hyaluronan, but many of them are based on hyaluronan. And then just in case that wasn't enough things to do with hyaluronan, now it's become a popular oral supplement. And whether it has any function in that way is probably controversial, but we can discuss that later if anybody is interested in that. Now, where do you get all this hyaluronan? Well, up until probably the last 15 years, where it's been supplied by fermentation, it used to be isolated from the combs of roosters and from the jelly that's inside the umbilical cord. It's called Wharton's jelly. And both of these are very rich in hyaluronan. Now people produce it by fermentation of bacteria. Streptococcus pyogenes, which is the cause of strep throat, has a natural hyaluronan coat, which is how it avoids the host immune system. And you can engineer bacteria to produce a lot of hyaluronan, and believe me, it is a big industry now, with the biggest amounts being produced in China, actually. So I've told you a lot about hyaluronan and how it's used commercially, but where is it actually in the human being? And it is spread throughout all of our tissues. It's in the matrix that surrounds our cells, called the extracellular matrix, or the ECM. And in this picture, it's depicted as this blue strand. And you can see there's other molecules in the matrix as well. It's actually a sugar polymer, so it's a linear polysaccharide made up of N-acetyl glucosamine and glucuronic acid. And if you remember some biochemistry about glycosaminoglycans, then you'll probably pick up that this is a member of the glycosaminoglycan family because it's made up of an amino sugar and a uronic acid. It's not really your typical glycosaminoglycan. Some of the more typical ones are like chondroitin sulfate and dermatan sulfate, but it is a member of this family. What's different is that it's so large, it can be 1 million kilodaltons. And it's also not linked to protein, which is probably no surprise because it would be hard to make something that big inside of the cell. So chondroitin sulfate is often added to proteoglycans, and you see a proteoglycan with chondroitin sulfate that is linked to the hyaluronan. So that's very common to see other proteins that are binding to hyaluronan, but itself does not attach to a protein. It is, however, very important in matrix assembly, and in some cases, if you actually prevent a cell from making hyaluronan, it won't make a pericellular matrix. So it really needs that hyaluronan to assemble this matrix. And maybe no surprise, after I told you about the Wharton's jelly and the umbilical cord having a lot of hyaluronan, the highest levels of hyaluronan are in loose connective tissues like our skin, like the vitreous body of your eye, the synovial fluid of your joint, the Wharton's jelly of the umbilical cord. All these loose connective tissues have really high levels. And it's also turned over in very large amounts. And I'll refer to hyaluronan as HA. It's actually one of the words I test members of the lab to see if they're going to fit or not, is whether they can say hyaluronan and hyaluronidase really fast 3 times. So that if they do, then they can work in the lab. So hyaluronan is turned over in really large quantities. Believe it or not, 5 grams gets turned over every day in a human being. And it starts locally where the hyaluronan is synthesized, and then some of it gets removed into the lymphatic system, shown in green. About 70% is removed there, and the remainder enters the bloodstream, where about 10% is removed in the liver. That is a lot of turnover. And it has to be synthesized and removed in large quantities. So here you see the main hyaluronan synthase. It's called hyaluronan synthase 2 or Has2. And this is sitting in the membrane, and it synthesizes hyaluronan from sugar precursors and extrudes it into the extracellular space. And as I mentioned, maybe there's some reasons why a supplement could have some impact on some phenotypes related to hyaluronan. And that's because the sugar precursors could be influenced by things you ingest. But again, I think that some of the work around supplements is pretty controversial. So what's made does have to be broken down, but first a little bit about hyaluronan's function. So here you're seeing just 2 disaccharide units of hyaluronan. Obviously, it has hundreds of disaccharide units. But what you notice right away is that these groups, the amino group and the carboxyl group that are provided by the sugars, are perfect for binding water. So you make all these hydrogen bonds with water, and so it is a very fluid-loving, hydrating molecule that cushions tissues and cells like to migrate in it. But that's not all that hyaluronan does. It also binds to receptors. One of its most classic receptors is CD44, which is involved in immune function. How hyaluronan mediates some of these signaling pathways is quite controversial because hyaluronan is a very big molecule, and some of these pathways seem to be activated by smaller molecules of hyaluronan. So this is where my own interest comes in, and that is in the molecules that break hyaluronan down into fragments, and they're called hyaluronidases. And they cleave within these large hyaluronan polymers and make shorter units of polymers. They're still big. And so hyaluronidase is a potential way to regulate the turnover of hyaluronan. And I first got interested in all of this because of, this classical love that I have for rare diseases. So I was working with a colleague in the United States. He was a clinician, and we had been working on lysosomal storage diseases like Tay-Sachs disease. And he said to me, I have this really interesting patient. She has mild short stature, and she has some problems like osteoarthritis of the hip is what it looks like. And she's got all these masses on various joints. And when they biopsied one of these masses and they looked at what was in the cells that were inside the masses, it turned out to be hyaluronan. So here we had hyaluronan that was being stored in lysosomes. So it was defining a new lysosomal storage disease, which was called mucopolysaccharidosis 9. And he said, do you want to try to figure out what the gene is that's causing this lysosomal accumulation of hyaluronan? And of course we did. This was before the whole genome project. So we looked for a gene and we found a hyaluronidase 1. And this is just a little pedigree over here. And I threw this in for some people who may like a little bit of enzymology in here. This is a gel that has been impregnated with hyaluronan. And when you run the serum from these individuals onto the gel, you can then take the whole gel and you can incubate it at the pH where it's optimum for the enzyme hyaluronidase to work. And then you can stain for hyaluronic acid, so for hyaluronan. And any place that there's no hyaluronan left because the enzyme has functioned gives you a clearing. And so this individual right here has no clearing because she has no enzyme activity. So that defined a new disorder from hyaluronidase deficiency. But what was really surprising was, how can you have such a mild disorder when there's so much hyaluronan that's being turned over every day? And this was something that created a bit of a conundrum for us. But right away, we thought there has to be other things that are breaking down this hyaluronan. So we began looking for other genes that were involved. And as it turned out, we and others found out that there were actually 6 hyaluronidase-related genes in the genome. HyLP1 turned out to be a pseudogene, so it didn't really matter. It didn't make a functional hyaluronidase. HyL4 broke down chondroitin sulfate, a different glycosaminoglycan, and not hyaluronan. SPAM1 turned out to be a hyaluronidase that is on the surface of sperm, and it degrades the hyaluronan that surrounds the oocyte to allow fertilization, but it's not expressed in any other tissues, so it wasn't going to be relevant in this large-scale turnover of hyaluronan. So we focused on genes HyL3, HyL1, and HyL2. And we made mouse knockouts for all of these genes. The first one, it was a weakly expressed gene, and sadly, the mouse had no significant phenotype that we could detect. So we moved on to HyL1. it had a phenotype very similar to what we saw in the human patients, and that left HYL2. HYL2 is broadly expressed, and the other unique thing about HYL2 is it's on the surface of the cells. So it's degrading hyaluronan that's outside of the cell, not inside of the cell. And that was very interesting to us. We proposed a model for the turnover, and in this model, HYL2 or hyaluronidase 2 would be sitting at the surface, and in cooperation with some kind of a receptor like CD44, we thought it would break large polymers of hyaluronan down into fragments, which then would be internalized and broken down in the lysosome. And we had already demonstrated that in the absence of HYL1, you did get accumulation of hyaluronan in the lysosome, but not that much compared to how much hyaluronan is present in an individual. And in fact, we were able to show that other glycosidases, beta-hexosaminidase and beta-glucuronidase, could compensate for HYL1 deficiency. So that somewhat explained the rather mild phenotype that was associated with a deficiency of hyaluronidase 1. And it really left us still a bit perplexed about what HYL2 was going to be doing if you had to have HYL2 for every aspect of hyaluronan degradation. So then we had our mouse knockout coming along. And one of the things that excited us about what we might find in this knockout was a piece of information that came in the literature about this time. And this was that the synthesis of hyaluronic acid is absolutely required for heart development. So hyaluronan synthase 2, as I mentioned, is the major enzyme that synthesizes hyaluronic acid, both in humans and in mice. And what they found was in a mouse at embryonic day 9.5, and mice have a gestation of 19.5 days. So this is pretty early in development. The mice would die. And when mice die that early, it's usually because their heart doesn't develop normally. So when the heart develops in a mouse, it originally develops as a long tube. And one of the first steps in forming the heart is for this tube to start to fold and start to get that shape of a heart. And when this happens, there are also swellings that take place. And these swellings are called endocardial cushions, and they're very rich in hyaluronan. And as it turns out, this cardiac jelly that forms these swellings that's rich in hyaluronan promotes a process where cells that are lining the surface of the tube change their behavior, and they release themselves from their neighbors, and they become cells that like to migrate and invade. They're called mesenchymal cells. And this process is called endothelial to mesenchymal transition, or EMT. And this is really important for development because this means that these cells, which lose contact with the other cells, can migrate, and they express new markers, and they can move out to form valves and other tissues. When you looked at this tube, so now we're looking at a cross section of the tube in a hyaluronan synthase knockout mouse, you could stain it for glycosaminoglycans, and that would be blue in a normal tube, and you can see the glycosaminoglycans are not there in the hyaluronan synthase knockout mouse. You could also stain for hyaluronan, which looks brown. And again, you would not see hyaluronan that was very abundant in the hyaluronan synthase knockout. And that's not surprising. And it turned out that these mice died because their heart didn't develop. But at the same time, they also showed that if you added hyaluronan back, then development could continue. But that was done using explants of these tubes. What was really disappointing was because these mice died so early, no one could really tell what else hyaluronan was important for. And we did think that hyaluronan would have other things that it did during development. And one of the reasons we thought this was because of a really nice paper that was created in the early 80s, where they looked at the hyaluronan levels in many different tissues in the developing mouse. This is an embryo at day 13.5. And at this stage, you can see that where the face is going to appear, there's a lot of hyaluronan. Anything black is hyaluronan. The vertebral column has a lot of hyaluronan. The developing heart has a lot of hyaluronan. The sternum, the gut submucosa, and many other places. So we thought that hyaluronan would be important in a lot of different tissues. Surprisingly, the mice that were deficient in Hyal-2 were not that severely affected when we first looked at them. And this was done in partnership with a group in Belgium because they had made the cell line to make the Hyal-2 knockout mouse, and they had brought it to Winnipeg, and we had made the mouse. But then we felt, since it was their line to start with, they should do the original characterization. So this was work that we did in partnership. And the mice seemed to live. They had some differences. If you looked at the length of the snout in the knockout versus the wild type, you could see that the snout was shorter and that the space between the eyes was wider. But that was about it. That was all that they saw in these mice. They did notice that some of them died a little bit earlier, but some of them also lived right to 15 or so months, which is a pretty long life for a mouse. So we were then decided, okay, well, now that they've had their chance to publish their paper, we would get busy and look at some of these mice ourselves. And we also noticed that they sometimes were sick. So we dissected one of these mice that looked very sickly. And we quickly found that their hearts were not normal. So this is, I should have had a little warning before this, right? These are not pretty pictures, but here's a normal heart in a mouse. And this is one of the hearts that we first saw in a knockout mouse. And believe it or not, this is the ventricle. And this is a really swollen atrium, really enlarged atrium. And when we collected these hearts, and then we would section across the heart so that you could look at some of the valves in the heart. And we then took some of these sections and we stained them for hyaluronic acid. And then you can move to the right. And what you see is in the normal heart, this is a valve. Valves are full of hyaluronic acid, so that's what this brown is. And this is kind of what they look like. Remember, you're looking at a cross section of something that would be, going through many sections of the heart. They look very thin, full of hyaluronic acid. And then this is what we were finding in the knockout hearts. You can hardly even identify that it's a valve because it's so thick and full of hyaluronic acid and cells. And we could quickly understand that this was at least part of the problem that explained why these mice were very sick. And later on, we would go on to do functional studies of the heart. But while we were doing this and moving on to understand what was happening, we were assuming that this was a progressive phenotype where hyaluronan slowly accumulated in the heart. We weren't sure when it started. And while we were working on this, before I go on, I should mention we did show that hyaluronan, using a biochemical approach, also accumulated in the heart. And I'll leave it at that because I have another picture like this later in the talk. So we got contacted by an investigator in the UK, Dr. Andrew Cosby, and he had been looking at a very large kindred of Amish individuals who were segregating cleft palate. So the black boxes indicate people who were affected with cleft palate. And what they did, because they had this large group, is they were able to map the gene causing cleft palate to a region on 3p21 in humans that happened to be the same region where HYL2 maps. They also sequenced some of these individuals and controls and showed that a variant in HYL2, lysine 148 to arginine, completely segregated with disease. So they had reached out to us to say, have you seen cleft palate in your mice? They had read our paper with the heart abnormality. And we really hadn't seen cleft palate in our mice, but perhaps some of you might know why you wouldn't see cleft palate in mice. And that's because in humans, we intervene at birth if a child is born with cleft lip and palate and help them to feed. But with mice, if they're born with cleft palate, they can't actually suck. And so they usually die really quickly after birth. And unfortunately, another warning ahead, moms will often cannibalize the pups if they're not healthy. So you may not notice that pups are disappearing from the litters. So we hadn't noticed cleft lip and palate in our mice, but we couldn't rule out the possibility that it was there. So was cleft palate deficiency, was HYL2 deficiency the cause of cleft palate? These are a couple of pictures of two of the kids that were among their affected individuals. So you can see this young boy has had a cleft lip repair. This individual had a cleft palate repair. And some of the things that you may notice is that they also have hypertelorism. That's the wide-spaced eyes that we also saw in the mouse. We also saw the broad nasal bridge in the mouse, and some of these children had hearing loss. So we decided, we did see these wide-spaced eyes in the mouse knockout, and just to remind you, see, we had the wide-spaced eyes and the short nose. So we thought, okay, what we need to do is actually look to see if their mice are being born with cleft palate and then dying. So this is a table that I'll walk you through. You don't need to pay any attention to anything except what I point out to you. So what we did was we took many mice that were heterozygous, so heterozygous female, heterozygous male, and we crossed them, and then we would look at the genotypes of the offspring. And we started at day 21. 21 days is when we normally separate the pups from their mother, and we get samples to genotype. So at 21 days, we found that of 169 mice that were genotyped, only 16 were HYL2 knockouts, and 60 were HYL2 wild types. The knockout and the wild type should match. So let me just, I'm going to just pull up one more thing here. If you look at a Punnett square, if you have heterozygous crosses, you should have twice as many of the heterozygotes that you do of the homozygotes, but the homozygotes should be equal in number. So in other words, if we had 60 wild types, we should have had 60 knockouts, but we didn't. So then we started looking back in time and carefully watching these litters as they were born. So we'd be going in constantly to try to count the pups that were born, and then the next day count them again in case some died and perhaps were eaten. And so we needed to be constantly counting these pups. And what we discovered was that actually about 1/3 of the pups died at day 1 after birth, and another third died between P2, that's postnatal day 2 and postnatal day 9. So there was really a lot of lethality. So the next thing to do was to look at some of these mice that had died and to see what happened to them. So here we have a picture. This is a micro CT image of a day 1 mouse that had just died. And it's colorized so that the denser the developing bone, the more yellow it is. Red indicates that the bone is not very developed. And the way you're looking at this is the image is coming right through the top of the head, and this would be looking through the front of the snout right here, and these bones right here are right on the top of the roof of the mouth. So you're looking from the top of the head down into the roof of the mouth and out the front of the snout. And perhaps what you'll notice is in the knockout mouse, there is a bone that really it looks to be almost missing. There's just a little bit of it left. That's the ethmoid bone. It basically sits right in front of the brain. This is a bone that was missing called the vomer. It looks missing or underdeveloped in this particular mouse. And then as you look down, you'll see other bones that are underdeveloped. Here's the presphenoid, which has a lot of extensions, and you can't even see them here. This is a close-up of the vomer. This vomer actually brings bones together right at the palate surface, and you can see the fusion and the wild type that's absent in the knockout. And this is another way of looking at the skulls where you use a stain that makes the bone pink and the cartilage blue. And you can see that wide space in the central cartilage, consistent with the broadening that we saw in these patients. And you can see the very wide bone that's present compared to the wild type or heterozygote mouse. And this is another picture where you're looking from the side. This is called a sagittal section. So you're looking onto, this would be, the eye and the nose area, and the lower jaw is missing. This is just the upper jaw. This would be brain back here. And I'm not going to show you all of the different bones, but what you should notice is in the knockout, there's a lot of space and there's areas that are not closed off compared to the wild type animal. And this is because they have what we call submucosal cleft palate. It's not a complete cleft in that the tissue is closed in the roof of the mouth, but some of the bones are not fully formed. And a really good example, this is a close-up. So now we're looking at a section right through face on. So this is a coronal section, we call it. And this is stained again for hyaluronan in brown. And this is that vomer bone that I was telling you about that was underdeveloped, that fused bones together. So this vomer should fuse with the palate shelf. And these are 2 bones in the palate shelf. And in the wild type, you can see the fusion, but in the knockout, the vomer bone is not fused with the palate shelf. But you can see that the palate shelf is very rich in hyaluronan. It's a bit thicker than it is in the wild type mouse. And this is an example of what we would call a submucosal cleft palate because everything isn't normally developed in the bone structures. We also tested to see if some of the problems in development could be shown in the by using a marker for developing bone. In this case, we used the marker Ostrix. And again, we're looking at a coronal section a little bit further back in the head. So this is a bone called the palatine process. It's an extension of that presphenoid bone that I showed you in the micro CT image. And you can see that in the knockout, it hasn't come close enough to fuse, and it's come much closer in the wild type mouse. And similarly, the cartilage here has positive precursors to bone in the nose, whereas you don't see any of that in the HYL2 knockout. And we've actually done quite a few studies of the bone, and it looks like they're underdeveloped, and whether these are just slower in developing or completely underdeveloped is something we haven't fully characterized. We also looked at hearing loss in these mice, and that was a little more difficult because we had to rely on looking at mice that had become adults, because we couldn't do these hearing studies in early mice before they were weaned. But of the mice that survived, they still had required a louder noise, so you had to have more decibels of sound at all of the different frequencies in order for the knockouts to hear the sound compared to the controls. So this meant that the mice did have a lot of similarities to the humans, and over time, many more humans have been identified with HYL2 mutations, and this is because of whole genome sequencing and whole exome sequencing. Patients get reported around the world, and we were able to look at these patients and compare their mutations with the phenotype. You can see that some mutations are more severe than others. There's some patients that aren't shown here that were actually stillborn, and this just indicates, you can have a pretty mild mutation, but you can also have some fairly severe mutations. And so this was interesting because, to me, the human patients had informed us about the mice. It had said, you need to go look at the palate. But our studies in the mouse really informed them about the humans. And they said, oh, we have to go to look at the heart because they've seen a lot of heart defects. And after looking at 17 patients, they found that heart abnormalities or cardiac abnormalities were even more frequent than palate abnormalities. And what they discovered was a range of defects that mostly affect the valves and the septation of the heart. A very interesting one that they found was sometimes there was cor triatriatum. This means that there's 3 atria. Here we see 2 left atriums in one human patient. And although we had seen lots of problems with the heart, we had never looked for cor triatriatum, and that led us to go back to our mouse model and say, can we use ultrasound and see this example of 3 atria? So I know I can't make this work, so I'm going to try to show you the picture of these atria. Can you guys see that?
Yeah, we can see the video.
Okay, great. So you can see in this video 2 left atria, 1 right atria, a right ventricle, and a left ventricle. So that was pretty exciting. We actually saw that in 40% of our mice, something that we hadn't seen without that information from the human condition. We also went back to look at skeletal phenotypes in our mice because of what was found in the human patients. So in the human patients, about 50% of them had this indentation in their chest that's called pectus excavatum. So we began doing the staining that I told you about, where you would take the whole skeleton of the mouse and you would stain cartilage blue and bone as pink. And what we could see in some of the mice, there did seem to be an indentation in what would be the mouse equivalent of the sternum. And there were a lot of bony abnormalities. There were ribs that were curved. There's ribs that don't align with each other. The manubrium was sometimes broader, and the xiphoid, which is at the very base of the sternum, was often enlarged. And this is work that I still hope to pull together and publish, but at this point, We haven't got that together yet. So it was one of those cases where I really thought that the human and mouse studies were so complementary. And there were a few things where we found things in the mice that were never looked at in humans, such as a missing kidney, whereas in the humans, they looked at the eyes and saw myopia, and we didn't determine that in the mice. But I think it was overall a very informative study. And I'm going to keep going because my time is getting short. We did do enzyme studies to show that the mutations either prevented the expression of these proteins, so you would see wild-type protein, sometimes no protein made, but in some cases protein was made, and we then had to revert to other methods to demonstrate that they really impacted HYL2 function. So in this case, we actually looked at whether the protein could get to the surface of the cell by using immunofluorescence. This is a permeabilized cell that's been transfected with wild-type HYL2, but if it's non-permeabilized, which means that you're looking only at what is on the surface of the cell, we could still see HYL2. But with many of the mutations, although protein was made, it would never get to the surface of the cell. So there was dysfunction. The protein, for whatever reason, was not able to get to the surface of the cell. There were two exceptions, and we were able to confirm that this protein really did get to the surface of the cell in other ways. So I'm going to skip that because time is moving along, and I want to do a little bit of studies for comment on why we think HYL2 causes this abnormal development. And so there are methods where you can look at the endothelial to mesenchymal transition by taking a piece of that developing heart tube and placing it on the top of a collagen gel. If you do this at embryonic day 9.5 in a mouse, then that endothelial to mesenchymal transition will take place on the surface of a collagen gel, and the new migratory mesenchymal cells will actually migrate into the collagen gel, and you can look at this process. And this had been looked at before, and using this, they were able to show that hyaluronan was absolutely required for endothelial to mesenchymal transition, but so was the breakdown of hyaluronan. If you didn't break hyaluronan down, then you didn't go on to form the valves. So you could make mesenchymal cells, but they couldn't differentiate into the cells that were required to actually form the valves. So we thought, is HYL2 required for this breakdown to stop endothelial to mesenchymal transition and promote differentiation? So this was our hypothesis, was if you don't make hyaluronan, the mice die. But if you make hyaluronan and you don't degrade it with HYL2, maybe you get too much EMT, you don't differentiate, and you get abnormal development of the heart. And we knew that there was lots of hyaluronan that was accumulating in the heart, and it was higher in molecular mass. And I've already showed you that there was lots of evidence that this was true in different tissues of the developing mouse embryo. We even saw that in HYL2 deficient hearts, adult hearts, there was sometimes valve-like tissue that was not appropriately placed in the heart. And this is what you see, this brown staining with a valve-like tissue actually down the ventricle of the heart. And we used markers of mesenchymal cells. So these are those cells that are now migratory. They are spindle-shaped. They don't like to adhere to each other. So vimentin is a marker of those cells, and we saw there was more vimentin in the knockout mice than in the controls. And we saw that this was true in the atria and in the ventricles. we could go to earlier stages of development and show that there were more Vimentin positive cells. So here we are at embryonic day 18.5, but I'm going to skip to this one because we were even able to show at embryonic day 14.5 in the mouse that if you used a marker of mesenchymal cells, there were more mesenchymal cells in the Hyal-2 knockout mouse than in the wild type, both in the atrium and in the ventricle, and that there was less VEGF, and that's the molecule that you need to produce to promote differentiation. And there was less of that in the Hyal-2 knockout than in the wild type. So we did some of those explant studies, and this is looking at a cardiac cushion that has been removed from the developing tube at embryonic day 9.5, the developing heart tube, and placed on a collagen matrix. And then what you're looking at is cells that are migrating out, and they're spindle-shaped because they're those mesenchymal cells, and the green is Vimentin. This is a wild type, and this is a knockout, and you can see it looks like there are more Vimentin positive cells in the knockout. And we were actually able to show that there were more Vimentin positive mesenchymal cells that were migrating out from Hyal-2 knockout explants compared to wild types. And we were able to show this at different depths. And I'm just on my last couple of slides because I know we're running out of time. But I wanted to show that although our mechanistic work has been in the heart, we have some evidence that maybe this is true in other tissues. So here I mentioned that we had the manubrium that sits right at the top of the breastbone. So this is a cross section of the manubrium stained for glycosaminoglycans in a wild type mouse versus a Hyal-2 knockout mouse. And although we haven't done studies to really solidify this, it looks like there are more cells in the knockout, indicating this idea that there's more formation of mesenchymal cells. And as these cells, which are called chondrocytes, start to mature, they become empty looking. So they get this white look. So you can see less maturation in the Hyal-2 knockout compared to the wild type, which is consistent with this idea that you form more mesenchymal cells, but they're not able to differentiate the way they are in the wild type mouse. So just to summarize then, I hope that I've convinced you that in the absence of hyaluronidase 2, that hyaluronan is actually increased in abundance and size. That's true in mice and cells from mice, but we've never been able to get human tissue biopsies to confirm that in humans. We also saw that many developmental abnormalities occur in Hyal-2 deficient humans and mice, and they seem to be in places where hyaluronan is really high in abundance. So these abnormalities do seem to be associated with tissues that have high abundance during development, at least in mice. And I think we have good evidence that there's increased numbers of mesenchymal cells in the heart, perhaps the bone, and that there's decreased or maybe abnormal timing in the maturation of these tissues. And so we believe that HA degradation by Hyal-2 may be required for normal differentiation. And finally, I'd like to close by expressing my gratitude to the people who do all the work, because honestly, I don't think there's a single piece of data in there that I produced with my own hands. This is all done by the people that work in my lab with the help of the funding agencies and the many collaborators that were involved to help us do those different ranges of technologies that we have. And I think that brings me to a close. Thanks everyone for your patience listening, and I look forward to answering any questions you might have.
Thank you very much for that fantastic talk. So I think that we are going to be taking questions from both the room itself and hopefully online there will be questions there also.
Sounds good.
Mishmi has a question online.
Yeah, just go for it.
Go for it, Mishmi.
It's me.
Hi.
Hi, Barbara. So amazing talk. My God, I learned so much, with collaboration and stepwise progression, finding the mutation, development. It's overall, oh my God, such an amazing work you have done. Of course, my interest is in the TME and extracellular matrix, where
Right.
and you have already shown that VEGF has a role in playing. So I'm just curious to know that that my job, I know, doing research, but is at what extent, because so much of use of HA for cosmetic purposes, anti-aging perspective, but at the same time, some of your work is showing that, sometimes it is too much of formation is the problem,
Mhm.
So where we are right now on the knowledge base with respect to presence of HA in the extracellular matrix and in the context of cancer, because I could find two specific cancer really this being studied. One of them is definitely breast cancer. So where do we stand? Is the presence or absence or more of the HA2 mutation is linked with the cancer progression?
Mhm. So in general, more hyaluronan is believed to be worse for cancer progression because it makes the environment more fluid and cells are able to migrate and grow faster. So that's the general finding. That being said, whether breakdown of hyaluronan is good or bad seems to be cancer dependent. And you can't universally say that if you have more hyaluronidase that it is good or bad. It's had mixed outcomes in different studies that have been done. From what I have seen, so there's a lot of work that has been done by commercial groups who have injected hyaluronidase for many different purposes. So hyaluronidase often gets injected in combination with antibodies and things to increase their uptake. And nobody has seen any detrimental outcomes from doing that. So even though you're producing a whole range of small hyaluronan products, when that happens, there doesn't seem to be any indication that that increases your risk for cancer. So I think that hyaluronidase's role during development must be highly regulated. And I don't think that having small fragments of hyaluronan or high molecular weight hyaluronan that's injected cosmetically probably increases your risk. I think it must be closely associated with receptors, and most of these injections are just hyaluronan that's not closely associated with the surface of cells.
Right. Yeah. Thank you. I do have one more question for the mice experiment. Can I ask that, Mike? How early, especially where the deficiency is the problem in the mice model, have
Yeah.
you tried, at the staging as you have done at the embryonic level, have you tried putting it back, like injecting it and fixing the problem?
Yeah, so we had this idea that if we could add this inhibitor of hyaluronan synthase, so there's a well-known inhibitor called hypomicron. It's actually 4-methyl-emboliferol. Some of you who work with enzymology may know that it's classically added to substrates to create this fluorescent signal. but it's a known inhibitor of hyaluronan synthesis. And so we did try to give that once to our mice and to see if we could restore normal development. But, you know, we only did it once and it didn't work out. And I tried to propose that as part of a CHR grant and it didn't fly. So I haven't been able to complete those studies, but the treatment with hypomicron is actually now something that is being done I think as a tentative cancer therapy to decrease the production of hyaluronan in tissues.
Wow, amazing. Thank you so much, Barb.
Yeah, no problem. Thanks. It was great to meet you today.
Yeah, same here.
Hopefully we have classroom student questions.
All right.
Yeah, Genevieve?
Hi, so I'm actually presenting your papers in one of my classes, and I'm trying to make like a map of all the different families, but it just says Amish. Do you know like where the Amish population was from in the world?
I do. Those particular patients were all from Ohio. So they're close to Cleveland, Ohio, and there's a Amish community, an old Amish community there, and the patients all came from there.
Yeah.
You're welcome.
Oh, we've got Courtney in the back here.
Thanks for the often talk, Dr. Triggs-Raine. I was just wondering, there's so many cardiac problems. Do those happen to like worsen with age, with IL-2 deficiency if there's like a buildup of hyaluronan that happens?
Yeah, I would really like to know that. I suspect there would be a buildup of hyaluronan, and I wasn't able to get any tissues from those families because the people in the United Kingdom that connected with the families in Cleveland didn't build a strong relationship, to be honest, with the families. And we were left with no way to access those families so that we could get any tissues to study further. Too bad.
Thank you.
Thank you.
Following up as maybe more of a comment on that, I think this has been one of, like, as a note to the students, this, I think, has been one of the keys to Dr. Triggs-Raine's extraordinary success, that she has really gone out of her way to forge very personal relationships with a lot of the communities that she's worked with in terms of rare diseases, right? Especially the Hutterites and the Mennonites and so on in Manitoba. You know, kind of not this concept of studying the population, but studying with the population, right? It's subtly different, but it's a big difference. I think we had another question.
I do have a question. So I'm curious about what are the current treatment options? Is this something for like CRISPR and other kind of things that I hear in the news but don't know much about?
Yeah, interesting. I had wanted to test hyaluronidase. So there is hyaluronidase that's made and commercially available. The sperm hyaluronidase is actually made as a soluble fragment, and you can purchase it, and it's FDA approved for treatment in humans. So I thought it would be interesting to inject that into the mice and see if we could correct the phenotype by injecting it really early. But we haven't been able to do those experiments. Just couldn't pull everything together to make it happen. But I think that would be a good potential treatment. Anything where it was a CRISPR-Cas9 would be interesting, but you'd probably have to do it really early in development to correct many of those defects, because you can see that the pictures that I was showing you of the heart, that's like embryonic day 9, that's pretty early in development in the mouse. So you would need to be doing that probably, you know, in the first trimester of pregnancy.
Is it typically screened for it during pregnancy?
You could do some kind of a probably a genetic screen to start with, especially in the populations that you know are high risk, like the Amish population. You could screen to see if they have the mutation. To my knowledge, we never saw anybody who had the mutations and had no phenotype. So that means that screening, you'd be able to predict that if you have these mutations, you're probably going to have some abnormalities. But as I said, you know, this is one of the disadvantages of someone who's coming in from another country and sampling patients that are in somebody else's country, because they didn't develop a relationship with the communities that then facilitated going back in and sharing that information and doing something productive with it.
Thank you.
No problem.
I'll do a kind of follow-up question here, just give the group time to come up with anything else. So, maybe like a two-part question. The first one I wasn't gonna ask, but I think Dr. Majumdar brought it up. So have there been any longitudinal studies in the children with the Hyaluronan deficiency, let's call it, in terms of looking at cancer rates in the long term? So that was gonna be one of my questions, but the original one was more, so you're presenting a lot of data with regards to the mouse data, right? It's wild type versus homozygous recessive, essentially.
Right.
But at least in the human population, is it that the heterozygotes have absolutely no presentation, or is it that we haven't looked?
I would say we haven't looked. There was probably no incentive to look, because again, all the patients were ascertained because they had cleft lip or palate to begin with, or they had some other syndromic illness that led for their DNA to be sequenced, but no one would have looked at the heterozygotes. And we actually do know that in the heterozygous mice, we did see some partial phenotypes. You may have noticed in the studies using the explants, where we had looked at the number of cells that were being produced, mesenchymal cells produced from the explants. There was some intermediate level in the heterozygous mice. And we also saw that when we were looking at the skulls themselves, there seemed to be some intermediate phenotypes in the Hyal-2 heterozygotes, but the phenotype of the mice was variable enough that it was hard to get any statistically significant difference. And so there seemed to be some genes that were moderating the phenotype. And I think that's probably going to be true in humans too, that you'll have some people that have a greater risk if they produce more hyaluronan, then they might have a greater risk of having a phenotype if they can't degrade it.
So the heterozygotes basically don't have enough of a difference to warrant investigations.
Nothing.
Right, okay.
Right.
Interesting. And in terms of longitudinal cancer rates, probably just no data.
Nothing known at this time. You know, and one of the major problems with a lot of these rare disease gene discoveries is that no one follows the patients after the gene discovery. And so I would be very surprised if we ever know what the cancer rates are.
Yeah, that sounds fair. Oh yes, we've got more questions from the audience here.
This is probably a quick one, but I'm just curious if you've done any work with like colonies in your region. 'Cause I'm from Cypress River, so I'm curious if you've worked in that area.
So there is an Amish population in Manitoba, but we haven't done any work on this particular disorder in the Amish population in Manitoba. All of the work that I have done within Manitoba has either been on Hutterite, Mennonite, or Indigenous populations, and it was always driven by rare diseases that we found that were more prevalent in those populations. And to my knowledge, nobody has presented in Manitoba from the Amish population with this disorder. I did go to the cleft lip and palate clinic at one point, and I was trying to see if they might be able to provide some samples to us. I don't think a thorough study was done, and you have to have a group of clinicians who are really interested in pursuing rare genetic disease. And because cleft lip and palate is often thought to have a complex etiology, so it's not thought that all of cleft lip and palate results from single gene mutations. So there hasn't been a lot of motivation on the clinical side to start sequencing individuals with cleft lip and palate for mutations. But I think that will come with time, and that we will eventually see that there's maybe even some less severe phenotypes associated with Hyal-2 deficiency. That probably didn't answer your question. I went all over the place, but no, I haven't worked around Cypress River. But I grew up not far from there.
Yeah, earlier in your presentation, you noted that some mutations in the Hyal-2 gene, basically, it allowed the protein to still stick to the surface of the cell, but I was just curious, like, have there been any studies on those particular mutations in comparison to others to see if there's more mild symptoms associated with those particular mutations?
Yeah, very perceptive. In fact, there are those mutations that allowed the protein to get some to the surface had a more mild phenotype. And the ones that had a stop codon that made no protein at all had the most severe phenotypes.
Okay, thank you.
Thank you.
Any other questions? Okay, well, in that case, I think we've had a longer than normal question period, which is awesome. So thank you to everyone, but especially thank you to Dr. Triggs-Raine for this fantastic presentation.
Thank you, everyone, for having me there, and I look forward to meeting some of you in the future.